I would like to speak to you today about vaccines, both as one who has studied and taught immunology for most of my adult life, and as a citizen of the world. My interest in vaccine acceptance was piqued when I found that some of my daughter's friends were reluctant to vaccinate their children. In fact, one of them contracted measles on her honeymoon in Italy, having not previously been vaccinated. At the same time, the Los Angeles Times published an online database showing the percentage of students with measles, mumps, and rubella vaccination at each elementary school in the Los Angeles area. And I found that the school to which my grandchildren belonged or attended had only a 65% prevalence of MMR vaccination. So after several years of thinking and writing about the problem of vaccine coverage, I came to understand it from several points of view. And the first is that we human beings are the most diseased species on Earth. And this comes not from some way of knowing the extent of disease for every species in existence, but rather understanding the epidemiology in recent human history. Our history over the last 2,000 to 3,000 years has included one epidemic after another. Epidemics that often killed 25% or more of the population in any given region of the world and lasted decades or sometimes even centuries. But even this is a great underestimate of the effects of epidemic disease on human history. Since the dawn of history, we have been continually plagued by what Jared Diamond called crowd epidemic diseases. Until the latter part of the 20th century, most families lost children to smallpox, scarlet fever, pertussis. There were birth defects caused by rubella. Occasionally children died from measles. And in other parts of the world, malaria still kills about 400,000 people per year, mostly children. This sort of epidemic disease was continuous for at least 2 millennia. Many more people died of these diseases than all the wars combined. And the question is why? We're exceptionally intelligent as human beings, but why are we as a species exceptional with respect to the loss of life due to infectious disease? And the answer is, in a very short span of time, we human beings completely changed our ecology. We evolved as small, dispersed communities of hunter-gatherers consisting of a few hundred individuals. But with time, we domesticated plants and animals, and eventually people became situated and lived in permanent communities of much higher density. At the same time, we sampled all of the infectious agents to be found in cows and sheep and horses and goats and birds, meaning we captured many infectious diseases from these animals, infectious diseases that were not evolved, or if you will, tuned to human beings. So how does this affect infectious diseases in the modern world? So this is a short version of what we call human disease ecology. And the first point is that pathogens found in low density populations are long-lasting and exhibit low or infrequent tolerance. That is, they exhibit low disease. You can imagine that this is because a pathogen that is too virulent would kill its host before it can be transmitted to the next host. And in low density populations, it takes a long time for diseases to be transferred from one person to another. Conversely, in high density populations, pathogens are able to be transmitted rapidly from host to host. And so there's no selection pressure to keep hosts mobile or even alive. And one metaphor for this could be a forest and the possibility of a forest fire. So if we consider an isolated stand of trees in a green meadow, this would pose no danger of a raging fire, just as an isolated band of hunter-gatherers posed no danger for a large-scale epidemic. Whereas for a dense, interconnected forest, one spark can ignite a holocaust, causing widespread damage that lasts a long, long time. Another point is that zoonoses are infectious agents that jump from one species to another And as their virulence is unpredictable because they haven't co-evolved with their host, zoonoses are very dangerous. And examples being Ebola virus, Lassa fever, hantavirus. They jump into the human population. Oftentimes they only infect a few hundred people or a few dozen people, and then they fade out of the population. And this was the case for Ebola virus until about 2014, where it evolved to be more infectious. And so now it can cause epidemics. And the concept is that we human beings are not only culturally connected or genetically connected, human beings worldwide are biologically connected in the present through our exchange of infectious agents and our common susceptibility to disease. We are more than simply individuals coexisting on the same planet. We are all hosts within a disease network of microbes and viruses. For example, as long as one person on Earth harbored variola virus, which is the cause of smallpox, billions of people, the entire population of the world, was at risk. Infectious diseases show the real-world consequences of individual actions on the community of humankind. So let's visualize this schematically. This is what the world looks like presently in the COVID-19 pandemic. Most people have not encountered the virus. Of the almost 8 billion people on Earth, most have not contracted the disease. There are people, the ones in red, that are infectious. And then there are a few people who have had the disease, and we hope they are immune. We don't know that yet, but we're hoping that this is the case. Now, one way that this could end would be after a decades-long pandemic, in which you might argue you could encounter herd immunity. That is, most people have had the disease and are now immune, and the disease starts to die out. Of course, it doesn't die out completely because there's always an influx of susceptible new hosts in the form of newborn children. And so this is how diseases typically go from being epidemic diseases to being childhood diseases, where most everyone gets the disease as a child and either dies or becomes immune. Now, just yesterday, the Director General of the World Health Organization was quoted as saying, "Never in the history of public health has herd immunity been used as a strategy for responding to an outbreak." So rather than waiting for a decades-long epidemic for this to decrease, we need a vaccine, and we need an effective vaccine. So let me just go over very quickly what the types of vaccines there are and what types are being developed for COVID-19. The three types of vaccines that are currently licensed in the United States are whole inactivated virus, live attenuated virus, and subunit vaccines. The whole inactivated virus is similar to that was developed by Salk for the polio virus. And it is a virus that is grown. It's the wild type virus, but it's inactivated chemically, usually with formalin. On the other hand, a live attenuated virus is an actual live growing virus, but it's been selected so that it doesn't cause disease. And examples of that are the MMR virus, the Sabin vaccine, the vaccine for shingles, vaccines like that. And the third type is the subunit vaccine, in which you make a protein that comes from the virus, usually by recombinant DNA techniques, and you add to that something called an adjuvant. Now, what's been occurring over the last few years, and now it's been accelerated at warp speed, we're told, are the development of new types of vaccines. And one of them is to use recombinant viral vectors. These are vaccines in which a innocuous virus, perhaps adenovirus, has been genetically modified to encode one little part of the SARS-CoV-2 virus. And so an individual would be infected with the adenovirus and make an immune response that would include an immune response to this a portion of the COVID-19 or the SARS-CoV-2 virus. Another is to use DNA or RNA directly that's encapsulated, usually with lipid nanoparticle. This type of a vaccine then turns your own cells into a factory for producing viral products, and then your immune system responds. What all of these vaccines have in common is the ability to provoke an inflammatory response, which is redness, swelling, heat, a little bit of pain. And this is actually needed to initiate a production of long-lasting immunity. None of these agents are intrinsically toxic, but the temporary discomfort that they produce is the result of the onset of an immune response. When your arm is sore after an influenza vaccination, that's because your immune response is actually working. And finally, for this, we can again rely on Tony Fauci, who said that finding an effective vaccine to prevent the infection with SARS-CoV-2 is an urgent public health priority. So once we make a vaccine, how do we know it is safe? Well, there are a lot of means in place to ensure for this. And the first is that the Food and Drug Administration analyzes the clinical data to ensure that there is safety. And in clinical trials for vaccines typically include tens of thousands of patients, half of whom get a placebo and half of whom get the vaccine. And both the safety and efficacy of the vaccine under study is then analyzed. It's only when that is determined to be safe is it then released for general public vaccination. Most of the most of the potential for clinical adverse effects are found at this stage before it ever gets to general public vaccination. Next, the CDC, the Center for Disease Control and Prevention, and the FDA maintain a nationwide monitoring system called the Vaccine Adverse Event Reporting System, which allows anyone to report an adverse reaction, either online, by fax, or by mail. Probably the fax is a little outdated at this point, but by online or by mail. There's also a program that's carried on between the CDC and U.S. healthcare organizations that track the data from millions of people who receive vaccines and then are monitored for their health. And finally, there's the National Vaccine Injury Compensation Program, which receives reports of adverse vaccine reactions, studies each claim, and then decides whether or not there's the possibility that an adverse reaction occurred based on vaccination. So for example, from 2006 to 2018, over 3.7 billion doses of covered vaccines were distributed in the United States. There were 7,400 petitions for adverse reactions, and 5,000 were compensated. So about 1 in a million vaccine dose results in a compensated adverse reaction. For measles, mumps, and rubella, there were about 108 million doses administered, and from those 108 million doses, 259 adverse reactions were filed and 128 were compensated. Again, about 1 in a million doses. It results in an actual adverse reaction. So let me close by saying that our modern, interconnected, densely populated world is only possible because of large-scale vaccine programs. The difficulty is that as we approach herd immunity in a region, everyone is protected, and thus there's a tendency to feel that vaccination is unnecessary. But I want to say that we remain protected only if the vast majority of the population realizes the importance of community participation in our common immunity to disease.
There's been some reports about an old malaria drug that a lot of people have gotten behind, but the test is not necessarily acceptable. I know it wouldn't be acceptable to a great vaccine company like yours. Can you explain the difference between what you're doing with Kevzara versus the anecdotal evidence that the old malaria drug might have some play here?
Look, let's take a step back, right? So you asked about the vaccine. We've got 2 vaccines in play for COVID-19, different mechanisms, one a more established mechanism and one a more experimental mechanism. I think it's incumbent upon us to make sure we have 2 shots on goal. it's important to get there as fast as possible in case, of course, there's a second wave. Then you've got the medicines that we're making every single day, the patients that need them, just to continue in their chronic therapies in asthma, in other diseases. The two medicines that are in the middle there, Kevzara and Plaquenil, which is a malaria drug, these are where people in China and other places have said, we've seen some effect. We have tried them and we can see some efficacy. Now, what we need to do is working with the WHO and the regulators is actually quantify that effect and quantify the actual patient profile that will respond best. We don't want patients struggling. We know that if repurposed medicines can be put to the task to do something incredible, then so be it. So we're just playing our part, making sure they're available to the investigators, making sure the regulators have everything they need.
But it does seem that your acute drug that you just described, the acute vaccine, it seems like that if we're really lucky, it can both take care of people who already have the virus in them and actually stop the virus coming in because of a very, a brilliant way. It's a brilliant way to be able to head off the virus's most insidious mechanisms.
Yeah, I think ordinarily it takes 10 years to invent a vaccine. The main reason is because you're going to give it to a lot of healthy people on a massive scale. So you have to be sure and you have to be right. These are, of course, unprecedented times. So we're experts in this and we're moving as fast as we can. And the science is going to tell us whether we can be in patients that need vaccination, patients that will be protected, or even patients that may respond who already have the illness. But we need a bit of time. And we're going, literally, I said, we've got everybody in the entire organization working 24/7, bringing their volunteer spirit to make sure we can do something with these vaccines.
In New York, another milestone. 2,000 cases in one day for the first time in 5 long months. And the doctors tell us it's about to get much worse. Everyone on Earth is waiting for a vaccine to cure what ails everyone and return us to something like normal. But what if that never happens? Hear me now. There's a British scientist who's now reporting COVID is likely to become not a pandemic, but an endemic. In other words, something that comes back every year for years and years and years, something we'll have to just deal with, like the flu. Dr. Amish Adalja is with us. He's a senior scholar at Johns Hopkins Center for Health Security. Doctor, you raised this possibility of coronavirus as an endemic, a recurring thing like flu back in February. Are you now convinced, or where are we?
I am pretty convinced that this is going to be something that we're going to have to contend with for years to come. It will be something that we sort of defang with treatments, with a vaccine that probably prevents you from getting severe disease. But it is going to be something that was probably going to follow the pattern of the other 4 seasonal coronaviruses that cause 15 to 25% of our colds every year.
And we just deal with it and ad infinitum, and do we continue with masks and just sort of move on in a different way? Does our economy figure out a way to recover? I mean, there's a lot here.
I do think what's going to happen over time is what people are going to have is a change in risk perception. We're going to have new treatments. We're going to have a vaccine that maybe doesn't prevent you from getting infected, but prevents you from getting hospitalized. And I think that will get the public health urgency off of this over time. I think for the first year to 2 years, we are going to be probably aggressive. People are still going to be advocating some degree of social distancing and face coverings. But over time, with new treatments, it is going to be something that becomes more like influenza. And maybe we'll get a second generation vaccine that'll be more like a measles vaccine and can drive it down to a low level. But I think we have to look at the pattern of coronaviruses and how they've established themselves in the human population and really think about endemicity becoming something, but with a lower virulence level, because we are getting better every day at treating this infection.
We are getting better, thankfully. Dr. Amish Adalja, thank you so much.
The FDA has approved Moderna's coronavirus vaccine candidate for a phase 2 trial. That's something that the company calls a crucial step in its timeline. Joining us right now with more on this news is Stefan Bancel. He is the CEO of Moderna. And Stefan, it's good to see you this morning. Thanks for joining us today.
Morning, Becky.
Let's talk a little bit about this news from the FDA. The stock jumped as it was reported that you'd be going ahead with the phase 2. In this case, you're going to be using 600 healthy patients, or healthy people, I should say, and injecting them with the virus. What does this mean for how that phase 2 works, and what does this mean for the timeline for when we might actually see a vaccine?
Yes, so this is a a phase 2 study, 600 healthy volunteers, to which we will inject the vaccine. The timeline should be pretty fast, and our goal is to be able to potentially start a phase 3 study with thousands of healthy volunteers in early summer.
Where do you find these volunteers? Who's going to be doing this?
I mean, there's always a lot of volunteers for vaccine studies, but especially in the case of the coronavirus, as you can expect, we're getting a lot of requests, and people want to help. People want to restart the economy, so there's a lot of people who are interested to participate.
And are those people who are related to Moderna? Are those people that you find who are hospital workers? I just wonder how that works. I mean, when it came to the polio vaccine, I think the doctor who invented it, his kids were some of the very first ones who were vaccinated with it too, because he was so sure that it was going to work.
Yes, and that was a different time. Today, that would be actually not legal to vaccinate people before approval that have a link to the vaccine development. So, for example, the employees of a company cannot be vaccinated. So usually it's just volunteers that either do many clinical trials, or in the case of the studies like you've seen on TV for phase 1 is just people who want to help and register onto clinicaltrial.gov. They have access to the clinical site, and then they get screened. They have several visits with the doctors, blood work, to make sure that they're healthy and that they qualify for the study.
So what does this mean for your timeline? I know that your next step was thinking that you would be phase 3 by the summer. When in the summer? Is that a June, July, August sort of situation?
So it's early summer, so it's still a bit too early to be more precise. Our previous timeline, Becky, was to start the phase 3 in the fall. So we're able, through a lot of good work and cooperation, to accelerate that timeline drastically. As we get closer, we will share that data. What I think is important is we should be able soon, and I'm talking weeks, not months, to know the human phase 1 data. And that will be important, not only for safety, which we anticipate to be similar to over 9 vaccines that have been in the clinic, but especially antibodies. Can we find in those healthy subjects the antibodies that bind to the virus and then neutralize the virus? That would be very important.
I know that this is a situation where emergency use authorization has been considered for this vaccine and for others, which is why it's happening faster. Normally, there's at least a year timeline before you would see an approval for a vaccination, just because they would want extended data to make sure that it's safe. What process would you kind of skip on that front, and what are the dangers by going faster? Obviously, we need a vaccination more quickly so people can get back to work and feel good about it.
Yeah, so as you say, Becky, the number one priority is safety, because you give vaccine to healthy people. You want to make sure the vaccine is safe. In such a situation of a pandemic where we have a lot of people that are sick and we are losing a lot of people, as well as the economic impact, speed is very important. So how do you balance the safety and the speed? Most probably for this product, if it gets approved by the FDA, you will expect that there will be a lot of follow-up to follow safety even after launch, which is typical when there is an accelerated approval.
You know, Beck and Stefan, I know you know this, back with the polio vaccine, which you mentioned, Becky, that it was made in animal, in cell culture, and we didn't even know how to test for SV40, which was a simian virus 40, and it was contaminated with SV40, but that didn't hurt humans, luckily. It was an inactivated polio vaccine. I'm glad we don't have to do an inactivated coronavirus, Stefan, and you've got the technology to do like one protein that's on the surface. Have you done? You saw the Oxford vaccine worked in monkeys, in rhesus monkeys. Will you test like that? And is that? Are they close enough genetically to where you have that Oxford should have, the university should have a high degree of confidence that the immunity would be the same in humans?
Yes, it's a great question, Joe. So animal models in infectious disease are very important, and usually they are pretty good predictor of what happens in humans. We are currently running several animal models with NIAID, the division of NIH led by Dr. Tony Fauci. And we hope that soon, and again, I'm talking weeks, not months, to be able to publish in a peer-reviewed journal the preclinical data. And in infectious disease, you can do a challenge model where basically you vaccinate the animal and then you give them a very high dose of a virus. And you can see in a placebo-controlled way, can you prevent disease? And this will be able to share that data soon.
And are you going to have you started manufacturing the most promising of your candidates yet? And would you do that near term? And how do you make millions of copies of that? Is it PCR or something? How do you do it with small little stretches of messenger RNA?
Guys, so it's a great question, Joe. So basically, we have started making in our Massachusetts plants as much product as we can. We announced last week the deal with Lonza, where we are currently transferring the technology to the US site of Lonza in New Hampshire.
Right.
And we anticipate to start making product in Lonza as well in July. So we're just stockpiling as much product as we can. And the process, as you say, it's a cell-free manufacturing process. It's in water, it's using enzyme. And so that allows us to replicate and to scale very quickly.
Right. Thank you.
Stefan, there's been a lot of questions about how this vaccine would be distributed. What do you know on this front about if you do have a successful vaccine and it's approved and you go ahead with it? Who would get it first? How would that be decided?
Yes, that's a great question, Becky. And what we anticipate is if you look at the few manufacturers that are both in the clinic already and that have scale for manufacturing. And if you do the math, it's only a couple of companies. We will all be supply constrained for quite some time, meaning we won't be able to make as many products as will be required to vaccinate everybody on the planet. And so, for example, in the case of the US, we anticipate to work very closely with the government and with key medical officials to decide who gets the vaccine, in the first batch. And then when you have a next batch that is shipped to the government, how do we allocate it between healthcare workers, people at high risk, and in different geography where you have more cases?
Is it fair to say that this has progressed at your best expectations to this point, better than your expectations?
It has gone faster than my best case scenario back in January. When we started this on January 11, partnering with the team of Dr. Tony Fauci, we were hoping to get in the clinic in the summer. And as you can see, we are already with a green light from the FDA to be in the spring, in May, in the clinic for the phase 2. The phase 1 was March 16, starting dosing. And now we're saying early July, we might be starting a phase 3 in early summer. So that is a timeline that is beyond our wildest expectation. It has been because of an incredible collaboration with the FDA, with NIAID, and people in many companies working very closely together, working long days, working 7 days a week now since January for 3 months now.